Alrighty, guys. So this week we're getting into chapter 7, which is really taking a step away now from looking at the body and how it's communicating, and focusing on when that body is under attack, when there's outside invaders, foreign invaders entering that body. How does it respond to that new kind of stimulus? So we're focusing now on those pathogens, those invaders, those foreign cells that can cause infection in our body, and looking at this word antigen and its corresponding term antibodies, and trying to appreciate how those two things function and help that signaling in our bodies for how the immune system responds to these pathogens. And what a time to be talking about the immune system and pathogens being those foreign invaders, as we are in the midst of that crazy COVID-19 coronavirus. So coming to understand a little bit better how viruses work and can cause infection in the body, and the difference between viruses, which is what we are experiencing, and some of those other pathogens that can also cause disease. So, chapter 7 has a pretty specific focus. There's a lot more to the immune system that we will get to talking about, and finding out a little bit further detail, and using coronavirus as the obvious example to talk about. Um, so we will go on a little bit about it, and hopefully we're not sick of hearing about it by now, but it is just such a perfect example to actually use. While I think of it too, one of those resources that I was talking about a few videos ago, I said that there's a couple of things that I, for one, am watching and taking some information from. And if you are interested, there is a series that is really focusing on coronavirus at the moment and how that virus behaves and the molecular biology side of how the infection works. And it's a podcast, but they show it on YouTube. But it's called This Week in Virology, TWIV. And they have lots of episodes. It existed before coronavirus, but obviously now that's really dominated what they're discussing. So taking it to that next level and applying some of the basic things that we're learning about the immune system and viruses and other kinds of pathogens, and listening to some of that higher level stuff, it'll help your understanding if you feel like you're capable of taking a little bit more in. So hopefully a few of you will go and have a look at those resources. And if you stumble across something interesting that you think I would appreciate, chances are I will. So let me know about it. All right, looking at chapter 7. So starting out with 7.1. The two key knowledge dot points that we are really focusing on for this first little part. So we want to be able to appreciate an antigen as a unique molecule or part of a molecule that initiates an immune response, including the distinction between non-self and self antigens and allergens. They are as straightforward and simple a definition as they sound, so I'll come back to those ones in a minute. And then looking at cellular and non-cellular pathogens as a source of non-self antigens, and preventative strategies, including physical, chemical, and microbiological barriers in animals and plants to keep them out. That second part of the dot point is really covered when we start looking at the immune system itself. So I might mention it during this video, but the details probably won't be done until we're looking at 7.2. So we're kind of cutting this dot point in half. Okay, so the first thing that we need to establish is that there's two very similar sounding words, and you will definitely catch me out using the wrong one at any given time. But we want to make it clear at the start. There is a big difference between the word antigen an antibody. So antigens are these markers that we find on the outside of the cell surface. So when we were looking at the fluid mosaic model of the cell membrane, and there was a group that was assigned those extra little bits, including cholesterol and receptors. And then there was one that was called identification, something along those lines. And it was pretty vague in general, the way we left it at the start of the year. But now we're going to look at them in a little bit more detail. So when we talk about markers on the outside of cells that are used for cell identification, we're talking about antigens. We're talking about these molecules that can be found on the cell's surface. Antibodies, which we misuse or accidentally say the wrong term every now and then, but antibodies refer to these other molecules that are produced by the immune system that attach to and recognize antigens. So the antigen is what's on the outside of a cell. The antibody is the thing that's going to attach to it and recognize it. Okay, so antigens, these are the things that are found on the surface of cells. All right. Okay, I just went and found this diagram quickly to sort of illustrate what I'm talking about rather than try to use my hands to represent antigens and antibodies. So antigens would be represented with these little shapes that are on the outside of a cell, and antibodies have this more Y-like structure. When you're doing a little bit of research about antibodies, a common thing that I see is this problem with the way they represent the antibodies. So having one like this, where you can see the shape that is on either end of that Y structure, rather than the shape being this part of the antibody. It's a small thing, but it bothers me because it's not right. So having the shape on the ends is more accurate to show. But what you can see is we've got antigens on the outside of these red blood cells. And in your style lesson, you'll see that the ABO blood typing system is used to illustrate as an example of antibodies and antigens. So our antigens are on the surface of the cell, and these antibodies are things that at the moment we'll just call them molecules that are present in the body, and they have the ability, the capacity to pair with, to recognize a specific shape. So this part of their structure is all important, and if they have the right shape, they can bond to an antigen, and they can do a number of things. So we will go into a little bit more detail about what antibodies can do, but just the sheer fact that they can cling on to the cell means that they're flagging it as something that is foreign, and they can join cells together. So they can form a clot of cells because they can join to more than one. So this one, say, will join to one cell, and this one will be able to join to another cell. And in that way, they can form this cluster of cells that they're bonding to. All right, I won't go into all of the detail because there's a video for you to watch from Bozeman about this ABO blood typing system. But what you can see is that if you have antigen A present on your surface, you don't want antibody A. If this person had antibody A, that would mean that their own antibodies are recognizing their own cells, and that would cause lots of health problems, blood clots, etc. So the idea is that you have antibodies for shapes that are not your own. So that kind of covers our first point about antigens. There are self antigens and there are non-self antigens. So for a person who is blood type A, their self antigen is A. For a person that is blood type B, their self antigen is B. And so when we're talking about self antigens in humans, we are often referring to what we call the major histocompatibility complex, our MHC proteins, which are the the antigens that we find on the surface of our cells. Just to give us a little bit of a graphic, this is just a different PowerPoint that I'll use for the rest of this series, but I only want it for this one diagram. And just this one diagram here kind of represents it nicely. So MHC markers are genetically coded. I'll mention again in a second about what antigens are actually made out of, but these ones are genetic, so we can assume that most of their structure is a protein structure. And because they're genetic, they can result in an array of different molecules that are present on the surface of your cells. And this set of antigens can vary from cell to cell. So you might have some of them present on one cell type and not on another. But the reason that it's important to appreciate that it's a genetic feature is because it explains why, when organ transplants are needed, it's often family members that will be the closest, what they call a tissue type match. And this is what they're talking about, because a family member is more likely to have the same set of MHC markers as you because of that genetic link. So having self antigens on your own cells is important. If any non-self antigens are detected by your body, you likely have an antibody present in your system that will be able to recognize it as being non-self. So while most of the time we would like our bodies to recognize what is non-self and get rid of it, in the case of organ transplants, if a new organ is detected as being a non-self tissue, then that results in the rejection of that organ. So self and non-self antigens, pretty straightforward concept. Self is your own set of antigens, non-self, anything that is not your own set of antigens. Okay, so what antigens actually are? So antigens are composed of lots of different kinds of molecules, not lots, the four major ones that we talk about. Most of them are protein based. So that's one important thing, that most of, or the majority of antigens are protein based. But some are carbohydrates, some are lipids, and some are even formed from nucleic acid. So antigens can be constructed from all of those macromolecules, but most of them are proteins. Just on these MHC ones too, while I think of it, there's actually two kinds of MHC marker. So when we're referring to the ones that are your self antigens, that set that I was just talking about, they are what we call MHC class 1. There is another one, MHC class 2, which will come up a little bit later when we're talking about the immune system and how it works, but they do a very special job. So if we just go back a couple of slides. Okay, there's a couple of other words that we attach to antigens. So self and non-self antigens is one thing. We can also call antigens immunogens. So if it is an antigen that is stimulating an immune response, then it's called an immunogen. And allergens. So that's the other one that's actually mentioned in your key knowledge in the study design. And an allergen, guess what they do? They stimulate an allergy response. The allergic response is something that we will learn the steps of later on as well. But just for now, as long as you can recognize that an allergen is something that triggers that allergy response. Okay, so antigens we kind of have a little bit of an appreciation for. The other big one then is our antibodies. Just to give you a look at what the structure actually looks like with this slide, They are something that we will come back to when we start looking at 7.2 and the parts of the immune system. Um, but it is, as we represented before, that Y structure. And just to point out here, that is where that variable region that I was pointing out with the ABO antibodies is. So this part here you would have many different antibodies that are possible to be made by your own body, and this region here varies greatly in its shape. When we're drawing antibodies, just in diagram form, we use lots of geometric shapes. So an antigen is represented with a triangle, and the corresponding antibody will have that complementary shape of the V shape that would fit around that triangle. They're molecules, they don't technically have that geometric shape, but it's fine to represent them like that in a diagram. But there's an endless sort of shape that these can take on, a huge variety of shapes that can be made by antibodies, and some are better at binding to particular antigens than others, but it is always far more complicated than we make it here. Right, so that gets a lot of the terminology side of things out of the way for 7.1. And then the fun part where you guys get to do a little bit of research. So, as part of this style lesson, we are outlining some pathogens that we want you to find out some more information about. So, the second part of the dot points that relate to 7.1 is all about pathogens as a source of non-self antigens. So, it's what the immune system is made for, is what it's meant to do. It's meant to detect these cells that could cause disease, can otherwise impact the normal functioning of your body, um, to detect them and to get rid of them. So we want to know a little bit more about what these pathogens are and what are some need to knows about how they work. So the ones that you need some detail for. Okay, the textbook details all of the following. You need to know about them in varying detail. So you're going to have to, unfortunately, use a little bit more than just your textbook for this one, but your textbook should be your first port of call. So read there, and if it's not giving you all the information you need, then you're going to have to do a little bit of extra research, but I'll give you a few little tips. So bacteria, just to point out first, they are separated into what we call cellular pathogens and non-cellular pathogens. So cellular pathogens, the best definition I can give you is cellular pathogens have the ability to reproduce without taking over the machinery of a host cell. Some of them require definitely a host to actually reproduce in, and that is part of their life cycle, but they do themselves replicate. So they don't have to take over the machinery of a host cell. Cellular pathogens are considered living, whereas non-cellular pathogens are not considered living. So we refer to them as non-cellular. And these ones, they cannot create copies of themselves without taking over a host cell to do that dirty work for them. Okay, so by far, actually, I might just, I'll quickly read through the list. So bacteria, we need to know a lot of detail about, and I'll point out a few things that you need to include in your summary. Fungus, there's a couple of key things that you need to mention. Oomocytes, read about it in your textbook. It's not part of the table, but have an understanding of what they are. Protozoans is basic detail. I'll point them out in the table. Worms is the other one from the cellular list that we've included in that table, just to have a little few facts about. And arthropods, more parasites, but I haven't included that in the table. I don't really ever see them asked about in exams. Viruses, viroids, and prions as examples of non-cellular pathogens. Definitely, they are all they're all key ones to know about. Viroids and prions are hard to research, so bare basic details just to be able to recognize what they are. And viruses, you need to know a fair little bit of detail about. So by far, bacteria and viruses should be your more detailed summaries here. I'll just open up style so we can have a look at what you're being asked to do. So for style, you've got a pre-test to do this week as well. Have a go at that one. It's a pre-test, so you don't need to know the answers, but it's good to do just as an intro. And here we go, working through your style. So the PowerPoint that I've been using to talk to through this, you can download here. Multiple choices. It's the video I said I wanted you to watch. Really just want to point out what you need to do with this table. Okay, so once you've watched all the videos and gathered a little bit of detail about these different pathogens, you've got this research table to fill in as part of your style for this week. So what we need, so I've put the first two at the top here as the ones that you need the most detail for. So by far in exams and just content-wise, you need to have a bit more of an appreciation of what this one is and what this one is. So I'll give you the heads up, those two are bacteria and virus. For these ones, we want to see some good details here. So name the pathogen. Is it cellular or non-cellular? They're one word each. List some examples of the pathogen or diseases that are caused by the pathogen. So for bacteria and probably for virus too, a lot of the infections or diseases that are caused are actually just the name of the bacteria. So listing off a few common ones. Describe how the pathogen reproduces. That's a really important one. So there's key terms that you need to be using in here. For bacteria, I want to read the word binary fission. For virus, you've got two that you need to give a little bit of detail for. So this one is called a bacteriophage. It's called bacteriophage because they are viruses that infect only bacteria. So that's some key detail that you should include. And this could be influenza, run-of-the-mill virus. But I want to know what are the parts and how does this virus gain entry to a host cell and make the cell do its bidding. So there's some details that you need to read through and summarize in your own words. And then treatments. So this is probably a short story, but you'll need to name what kind of treatment you would use for a bacteria, and it's one that commonly people think can be used for both, but is only effective against bacteria. And what does it actually do? So how does it cause the bacteria to malfunction and kill it off? And for viruses, is there a similar treatment that we can use here? All right, you've got a little bit of fun, I think, in identifying what the rest of them are based on what you read about them. What other details can I give you? So for the remaining ones, it's really, especially for these two, being able to look at the diagram and recognize what it is based on its structure. That's probably the most important thing that you need for these. So short detail for the rest of it. For these ones, you need to make sure when you're researching, I think especially for this one because it's not covered very well in the textbook, you need to make sure that when you type in the pathogen name, you include the word pathogen with it. So one of them is worms and one of them is fungus. If you just type in worms reproduction or fungus reproduction, you're going to get a lot of detail just about the fungus growing in your garden or the worms that you find when you're digging in the garden. So make sure that you type in the word pathogen to get some specific information about how they're causing infection. When you're talking about fungus, I want to read the words spores and hypha. So they're important things to understand for a fungal infection. But fairly basic detail for those ones. All right, so hopefully that makes the table a little bit more clear and what you're being expected to do there. And then you can have a bit of fun with the crossword. I have made a model answer for it that will come up when your answers are released. And it was a little bit time consuming to type it. There is a way to put a text box in and type the letters, but it doesn't have to be neat. So you can just use the hand drawing tool to draw over the top of it. Okay, best of luck, and I will see you during the week.